Hi, everyone. Thanks for joining us today. We'll get started in about 1 minute. Okay, hi everyone and welcome. We're a couple minutes past the hour, so we'll go ahead and start. I'm really excited to tee up our webinar today. I'm Connie Mitchell. I'm a scientific program manager at the Health Environmental Sciences Institute. And just to go through some logistics before we start today. So everyone other than the speakers will be muted. Questions will be typed in the question box. I'll have a slide on what that looks like. We will record the webinar and we'll share with registrants this week. We'll have time after the presentation for questions. And if you have any trouble, please email me at cmitchell@hesiglobal.org if you have any issues. And here's just what the question box should look like if you're either on the web or on the desktop app. So you can put your questions there and we will go through those at the end. So just before we get started, many of you should know, but HESI is a scientific not-for-profit based in Washington, D.C. We work with people from industry, academia, government, and other scientific organizations on topics you couldn't tackle in just one lab or even in one sector. The work presented today is going to take place within our Emerging Systems Toxicology Committee, and our mission and objectives are listed there, but it's really to use systems toxicology approaches in risk assessment. Here is our current portfolio. The work that we're going to be talking today is within our FFPE working group that Leah is going to walk through. This group has sunsetted, so this will be really a way to bookend that work and highlight it. So now to turn to our speaker, I have the pleasure of introducing Dr. Leah Wehmas, who is a genomics scientist with the Center of Computational Toxicology and Exposure at the US EPA, where she uses toxicogenomics, new approach methods, and systems-based biology to better understand the adverse effects of environmental chemicals and enhance traditional safety assessment. Leah received a BS in cell and molecular biology from the University of Minnesota Duluth and a PhD in environmental molecular toxicology from Oregon State University. After that, she was an R Authority postdoc fellow at the US EPA. Dr. Wehmas started working with HESI as a postdoc fellow in 2016 and served and led the working group applying a genomics to formalin-fixed paraffin-embedded tissue and has contributed to other projects, including our point of departure working group. She has authored and co-authored over 30 publications in the field of toxicology, 6 of which include archival tissue samples. One of her current efforts at the EPA is using FFPE tissues from the National Toxicology Program to understand species-related differences in transcriptomic responses to chemicals. So with that, thank you, Leah, and I'll turn over to you and let you share.
Okay, thank you, Connie.
Sorry, 1 second. Okay, you can present now.
Okay, and can you see my screen okay?
Looks perfect, thank you.
Okay, it's great. Thanks, Connie, for such a great introduction. And as Connie said, I'll be discussing the genomics from formalin-fixed paraffin-embedded tissue samples, which we'll refer to as FFPE, but the methods and lessons learned in risk assessment. There we go. This is my disclaimer, working with the EPA. And thanks again to Connie for providing a nice introduction to the eSTAR committee. And as Connie indicated, the FFP working group is really just one working group amongst many in the eSTAR committee portfolio. And this work will really cover all of the great work that we've done over the years involving multiple different people and groups. And that's kind of what this slide is highlighting and acknowledging some of our major contributors over the years, which they've done a lot of fantastic work and really helped advance the use of FFPE in genomics and risk assessment. And here are just a few names. I know this is not an exhaustive list, so I will be, I'll have a lot of the papers associated with the work cited, which will have a listing then of all the authors and all that information associated with everyone who's worked here. And of course, at EPA, I can't not talk about that a lot of this work was funded through the Chemical Safety for Sustainability National Research Program at EPA. And just as an overview of some of our major contributions to the field, this is just a summary figure of where we've disseminated some of our research findings. We've had, this is a plot based off of the area is proportional. So we've had lots of posters and abstracts at national scientific meetings, including the Society of Toxicology, Gordon Research Conference, Environmental Mutagenesis and Genomics Society, Society of Tox Pathology, as well as several regional scientific meetings that aren't listed in this chart. We've had manuscripts published in ToxSci, as well as Nature Scientific Reports and Tox Reports. And we've had invited presentations with the Society of Toxicology, including one really great roundtable session, as well as talks through the Health and Environmental Sciences Institute and talks with the Society of Tox Pathology. And for the HESI meetings, because of our really good level of productivity, one of our research was highlighted at the Dublin International Meeting for HESI in, I think it was 2017, where Dr. Susan Hester presented. And of course, we've also contributed to some best practice reports in collaboration with the National Cancer Institute and their biospecimen research program. And as Connie indicated in her introduction, I started this work as a postdoc with the EPA, and I was able to use some of this research to apply for the Next Generation Humane Science Award, which I received from Johns Hopkins University. So we've had we've been very productive over the years. This is just a diagram showing our publications since, and I know that this group started prior to 2016. 2016 is around the time when I joined the group, and that's kind of when some of the research started being published, but it's been productive even prior to that when it first started. And we've continued to produce publications over the years and be impactful in the scientific literature through citations. And we've really made an impact in identifying a lot of factors associated with using archival samples for genomics. And really what I want this slide to show is to act as kind of a overview of what I'd like to speak about today, highlighting some of our major impacts in the field and how they've built upon each other in a very logical way throughout the years. And so as I go through each of these bullet points, we can then, I'll describe in further detail the associated research and results from those major findings, and that we really helped advance the use of new technologies to get better data from FFPE. We characterize some of the major factors that impact their quality, identified biochemical methods to improve quality, identified better ways to measure their quality to determine whether you should sequence or not characterized artifacts, and also translated our efforts to clinical samples. So why would we want to do that with archival tissue samples? Well, as I indicated in the previous slide, one of the most attractive features with working on them is that a lot of the methods we developed can be applied for animal study related tissue samples or human clinical samples. And if you can get useful data from them, we can understand biomarkers and toxicity pathways, use them in clinical research to better understand disease development, and also use them in proof of principle type research for understanding risk assessment. With all without having to do new animal studies, which is also a key feature using archival samples. And in particular, why EPA was interested in working with archival tissue samples is as we, in the field of toxicology, as we move towards more higher throughput, short-term bioassays, there's really a need to link that information with the more traditional endpoints of toxicological and regulatory concern. And with archival tissue samples, we can get that sort of information and put it into biological context. And we can also use the adverse outcome pathway framework to really organize that information meaningfully. Moreover, there's been some efforts at EPA and elsewhere, including one of the other HESI working groups, that the point of departure group in particular, where there's been an interest in using dose response modeling of these molecular endpoints to set levels of concern that may indicate the 2-year apical adverse endpoints of toxicological and regulatory concern using benchmark dose modeling approaches. And what we've shown is that these short-term gene expression-based benchmark dose values are quite consistent with the apical endpoints of concern, and that this is just a publication by Gwynn et al. And highlighted in a publication by the National Toxicology Program looking at many different chemicals, where they related transcriptomic or gene set-based points of departure with apical endpoints from 2-year traditional assays, and that the gene expression-based benchmark dose value was within tenfold of the traditional. And so just step back 1 second here. And so what the HESI point of departure group is working on is really helping to set a framework for using that type of data in regulatory applications. And archival tissue samples in particular help us do those proof of principle type studies where we can relate that information within the same samples without having to do a brand new animal study. And some other reasons why we would want to work with archival tissue samples is that there's a lot of them around. This is why I think the archival or FFP group was started, is that there's billions of archived samples around there at places like the EPA, at the National Toxicology Program archive, they're in industry archives, and they often come from very valuable, expensive studies that won't ever be repeated again. They come with well-characterized pathological data or clinically annotated, and as the expense of doing some of these higher throughput gene expression technologies have gone down, that's really enabled us to rapidly assess target pathways, dose response, and translate information at the genomic level to human health relevant pathways so that we can identify candidate signals of toxicity from chemical exposure in a more efficient way. It's just that working with FFPE tissue samples has posed some significant challenges, and that's why their use has been limited for genomic applications to some extent, is because while formalin fixation works really well for preserving tissue morphology, it is not so great for the nucleic acid. The formalin fixation introduces adducts to the nucleic acid, crosslinks, and it can fragment the nucleic acid, which all can impact quality. It can be difficult to adequately assess the quality of the sample, and there's the potential for artifacts. So this can all lead to inconsistent gene expression results downstream. So what the FFPE working group thought is, what can we do to get better data from these types of samples? And what can we understand about those samples that impact their quality for getting useful gene expression information? And they really thought, I think, to use new technologies to help overcome some of these issues. And that's where our first two publications come into play where we applied RNA sequencing technology as well as two different library preparation methods to see if that could improve gene expression detection in FFP tissue samples, and also then to look at how time and formalin would impact the quality of the gene expression results. This was a very nice study where there was paired liver samples that were either fresh frozen or fixed for 18 hours as an ideal fixation scenario, and then a worse or case scenario where they were fixed for 3 weeks, which can happen with, especially with toxicology samples. And what you can see on the left is some concordance plots comparing the frozen gene response data to the 18-hour formalin-fixed samples across microarray, which was the established technology at the time, as well, and then poly-A enrichment RNA sequencing versus ribodepletion. And what we see here is that at least for 18-hour formalin-fixed samples, the RNA sequencing performed better. We get better concordance values with ribodepletion looking better than poly-A and the microarray. And when we go to 3 weeks formalin fixation, we start to see that there's a increase in degradation of the sample. So the poly-A enrichment RNA-seq is not performing as well, but the ribodepletion method is performing better. There's a better concordance in values. So time and formalin really does impact our ability to detect gene response in FFP tissue samples. But ribodepletion RNA sequencing can help overcome some of those limitations. But what about fixation time or long-term or really old FFP tissue samples? So that was another factor of concern. In these archives at EPA, we have old FFP samples. They have really old ones at the National Toxicology Program archive. Can ribodepletion RNA-seq help overcome any limitations associated with age and block? And in this study, we were very fortunate to have these two very well-studied model chemicals, di-2-ethylhexylphthalate and dichloroacetic acid, short-term exposures with multiple dose levels, where we had liver samples that were either flash frozen or fixed for 24 hours in formalin. And in this case, the unique factor especially was for the dichloroacetic acid samples, in that we did have 20-year-old paired and fixed tissue samples. So we could sequence all of these and compare them to each other to see how ribodepletion and RNA sequencing would perform with really old tissue samples. Moreover, because we had dose response information, we could look at how short-term gene expression response would indicate later adverse effects. Since both of these chemicals are known rodent tumorigens, we could compare their apical response data to the transcriptomic response, at least for the frozen, if not, hopefully for the FFPE samples. And what we saw is that the 2-year samples, the gene response and RNA sequencing data was really good. There was a high level of concordance and overlap in the 2-year fixed tissue samples relative to their frozen for DEHP across all the dose levels. This slide here is just showing some representative figures from that study, but they all looked really good. So we have good concordance. And when you look at the benchmark dose values of some marker genes of interest associated with DEHP exposure, ACOT1 and CYP4A10, the ACOTs have very consistent dose response effects that were modeled, and the benchmark dose values associated with those two genes were very consistent at 87 and 73 mg per kg per day, and it was similar for the CYP4A10 at 44 and 47 mg per kg per day, and these values were actually quite close to the 2-year adenoma carcinoma benchmark dose for DEHP, which I think was around 30 mg per kg per day. So they were quite consistent, so able to show in some proof of principle application that molecular data could indicate later adverse effects, and that 2-year fixed samples at least performed well for RNA sequencing applications or using RNA sequencing applications. But unfortunately for the 20-year-old samples, they just appeared to be too damaged to overcome any limitations. We still saw a little bit of overlap in significant differentially expressed genes in FFPE versus frozen, but very few. And really, I think it was only at, more so at the higher dose levels of DCA. And of course, there was more variability. Interestingly, though, if we focused in on some well-known marker genes from DCA exposure, in this case CYP4A10, the benchmark dose values from those two genes were actually still relatively consistent with each other despite all the degradation. So what it suggested to us is that perhaps with some of these really old samples, maybe if you wanted to learn something about a very abundantly expressed marker gene, perhaps you could get some information, but really 20 years seemed to be too much damage for RNA-seq, ribodepletion RNA-seq to overcome those limitations of those samples. So what we learned from those two first studies is that, again, time and formalin and agent block are significant factors influencing quality. RNA sequencing can help, but especially with ribodepletion methods, but that it couldn't overcome all the damage in those really old samples. So that led us logically to think about, are there ways that we can improve the quality of RNA from these FFP tissue samples? Are there methods we can employ during our RNA isolation that could reverse some of the damage that happens with fixation? And so that's where we were able to employ some biochemical methods to investigate how they might improve RNA quality. And we also were able to look at some other ways to measure quality in these samples. And so what we had learned is that there were a couple of procedures, either with tris acetate EDTA or incubation with this organocatalyst, that suggested that perhaps if you did this during the RNA isolation process, you could reverse or remove some of the adducts, reverse some of the crosslinks associated with formalin fixation in tissue samples. And so we really wanted to test this with actual samples. And so we had frozen liver samples collected from mice exposed to phenobarbital or vehicle control. And those samples were then split to represent this profile in degradation from 3-month formalin-fixed samples, so worst case scenario type of situation, to an 18-hour formalin-fixed, which is more of an ideal fixation scenario, and then to something that doesn't cause crosslinks but isn't necessarily ideal for morphology purposes, is a 70% ethanol fix. So that was really a control for the formalin fixation process. And then we could take those worst case scenario 3-month formalin-fixed samples and apply what we termed our demodification procedures, which included the organocatalyst as well as the tris acetate EDTA. So if you look at this figure, the DTAE represents DMOD for tris acetate EDTA, and the DQ and DP are the organocatalyst treatments. It's just we use 2 different kits with that procedure, and then the no D is the control for the organocatalyst. So it was really just an overnight low temperature incubation without the catalyst part. And so we could look at all of these together, performed RNA sequencing on all of these different samples, and look at how the fixation process and demodification potentially improved the quality of our samples. And so what we saw is that we looked at a lot of, this is pre-alignment effects, so not looking at the influence, at least yet, at the influence of the chemical treatment, but just at the samples themselves, pre-alignment from RNA sequencing results. We looked at a lot of different metrics. This is just one nice representation of the results where we're looking at read deletion rate across the sequenced reads, and what we want to see is a nice low level of deletion rates, so you're not seeing a lot of errors in the sequence samples. where the frozen and the ethanol are. So down at the bottom of the graph. Midway, we're seeing the 18-hour formalin-fixed samples. So fixation is causing some problems. And then up at the top, we're seeing the 3-month formalin-fixed samples. So more damage, poorer performance with higher redeletion rates. Interestingly, though, all of our demodification procedures seem to reduce the deletion rates in the samples, which would result in fewer potential errors for downstream gene expression results. So very positive, especially considering that these samples were derived from the 3-month formalin-fixed tissue samples. So now they're looking more like an 18-hour formalin-fixed tissue sample. And when we extended our results down to post-RNA-seq read mapping and differential gene expression analysis, we did see now that there was some added improvement of using organocatalysts. So I know this plot is very bit busy, so I'm going to walk you through it. It's a principal component analysis on our differentially expressed genes induced by the phenobarbital exposure. And so what you'll see on the left and right is that we see this separation based off of the chemical treatment. So phenobarbital left, control on the right, and then we see our shift in quality up the figures. So frozen and ethanol fixed samples are clustering near the bottom. Then we have our 18-hour formalin-fixed samples midway, and then our low quality 3-month formalin-fixed samples at the top. And our organocatalyst treated samples are sitting almost right on top, or clustered very tightly with the 18-hour formalin-fixed samples, whereas now the trisacetate EDTA and the catalyst control, the no demodified samples, are more in between. They're more variable. They're showing some improvement, but not quite to what we see with the catalyst, the consistency of using the catalyst. And this really did extend all the way down to overlap in differentially expressed genes and pathway level data, that the organocatalyst really did show some improvement and was able to improve the gene expression data from these 3-month formalin-fixed samples. So after that, we had already undergone, or we'd already performed RNA sequencing on a lot of samples that had varied widely in quality from their time in formalin to their age in block. And we really wanted to know, what we had seen is that the RNA integrity number, which is something that is traditionally used to determine whether you should do a QRT-PCR or microarray or RNA-seq, that it doesn't perform well for determining whether you should sequence an FFPE or sample where RNA was collected from an FFPE tissue sample. And because we could have a sample that had a RIN of 2 and it would sequence fine, or we could have a bad sample that had the same RIN and it would fail in sequencing. And so we wanted to know, we could retrospectively analyze these samples that we've sequenced that have these different levels of quality to assess whether other methods might perform better at indicating sequencing success. And so we looked at the RNA integrity number, the DV number, which is also referred to as fragment analysis, the PERM value, which is paraffin-embedded RNA metric, as well as some QRT-PCR-based methods, the more typical, as well as digital droplet PCR. And interestingly, the RIN, the DV, and PERM methods are all bioanalyzer based. So you would collect this information collecting the RIN value. So it wouldn't add anything extra to the workflow to implement measuring these variable or these metrics. And for the PCR-based methods, we looked at a couple of different housekeeping genes, different amplicon sizes, to see which might be good indicators of overall RNA quality in the sample. And what we found was that the RIN value, if you associate our RIN values with our gene detection in our various different types of samples, so age and block samples, 2 years to 20, or short-term formalin-fixed versus long-term formalin-fixed, that they just don't correlate well. As was indicated previously, you can get a good RNA integrity number and terrible sequencing results, or like relatively good RNA integrity number. And the same thing happened with the PERM, which it was designed to assess whether you should perform qRT-PCR. So we were trying to apply it to RNA sequencing, and that may be why it didn't perform as well as we had hoped it might. But the fragment analysis method worked quite well. And what it does is in the bioanalyzer, it measures the proportion of RNA fragments set higher than a certain threshold. It was originally proposed to use 200, so the proportion of your samples that have fragment sizes greater than 200 nucleotides. But we also looked at a couple of different size ranges with our samples to see, and we actually saw that while DV200 did work pretty well, the DV100 worked a little bit better in helping us distinguish which of our samples would have been good to sequence versus not. So if you look at the top graph, that 80% threshold helped us. If we had something like that, we would have only sequenced maybe 2 samples that wouldn't have been worth sequencing, and the rest would have all been pretty good for an FFPE tissue sample versus the DV200, which would have resulted in us sequencing a few more poor quality samples. And then when you look at the bottom, that's just a representative example of one of our housekeeping genes that we looked at. And each panel is a different amplicon size. So 80, about 127 or 429. And as you would expect, the longer the amplicon size, the harder it is to get, or you don't get as good of a correlation with the response, especially for these more fragmented FFPE tissue samples, or long-term formalin-fixed or really old. So it looked like a shorter amplicon size for the qRT-PCR or the digital droplet PCR-based method, around 100 base pairs in length, better associated with indicating quality. And so from this, we could really develop a strategy for sequencing FFPE samples where you could do an initial evaluation of the DV100, and if your sample had, most of them had DV100s greater than 80%, then it would be okay to sequence. If the DV100 was very close to 80%, then maybe you'd want to do qRT-PCR. And if you ran a few housekeeping genes, you'd want to keep the amplicon size around 100. And if you got about 500 copies of those housekeeping genes, then you're probably okay to sequence that sample. If not, then maybe you want to implement something else like the organocatalyst treatment to improve the quality, or perhaps not sequence that sample. So from this, we were able to use the organocatalyst to improve gene expression data from really long-term formalin-fixed samples. And we used fragment analysis to better characterize the quality for sequencing, as well as using qRT-PCR to help us develop that strategy to determine whether you should sequence or not. But throughout the course of this work, we had noticed something interesting with fixing the tissue samples, in that, and this is a principal component analysis from the 2-year DEHP study, is that when you fixed live tissue directly in formalin, which is what would typically happen when you're collecting samples, is that there's this shift in gene response that appears to happen that's reflected in the principal component analysis very consistently across the paired samples. And it led us to hypothesize that there's something about placing a live tissue into formalin that's causing this effect. And since we already had a study where we froze the samples and then fixed it, we could really investigate, is that really a true effect, or is some sort of artifact of fixation that maybe we need to control to better understand our gene expression analysis and downstream results. And so we were able to take our liver samples that were either directly fixed using a traditional fixation method, or they were frozen, and then divided the frozen up to those that were either frozen and then fixed in ethanol, which is, if you look down at the bottom, that'll be how it's designated, FR to OH, so frozen to ethanol or frozen to FFPE, or the traditional, which is represented as just FFPE. And of course, just straight frozen is just frozen, FR. And so we could then quite elegantly look at the impact of direct fixation on potential artifacts and gene response associated with that. And we had a chemical treatment built into the experiment to look at how it might impact our detection of the phenobarbital response versus control. And what we saw, like with the DEHP experiment, is that direct formalin fixation caused this shift in gene response. So that's what this principal component analysis is showing. The frozen is in blue, the directly fixed samples, FFPE, are in yellow, and there's that consistent shift across. And then if we froze the samples first and then fixed them, they seem to be a little bit more similar to the frozen, and that was what we saw when we looked at overlapping significant differentially expressed genes, is that if you froze the sample and then fixed, or froze in formalin or in ethanol, there wasn't much of a gene response, but there was a significant gene response if you directly fixed the sample in formalin. So we saw almost 3,000 differentially expressed genes, and actually a lot of them were downregulated versus if you froze the samples first and then fixed. And we were able to look at those genes and actually look at some other studies where we had directly fixed FFPE tissue samples for consistency in this formalin type of response. And when we mapped those two canonical pathways, we saw some effects associated with oxidative phosphorylation, mitochondrial dysfunction, and these are pathways that can be impacted by chemical exposure. So we just wanted to make sure that when we look at the chemical treatment response, that this isn't going to impact that. And fortunately, it didn't seem to. I mean, we still see some differences in the differentially expressed genes. Now this is looking at phenobarbital versus vehicle control in the directly fixed FFPE, frozen, then fixed, and then just frozen. And we're still seeing a pretty robust phenobarbital-like response. This is a well-studied chemical, so we see a lot of the genes known to be impacted by phenobarbital exposure that we were still able to detect in these samples. And if we map that response to predicted upstream regulators, we independently identify phenobarbital as one of the predicted upstream regulators. So this is positive for us because despite this formalin effect or effective direct fixation, we're still able to detect our chemical treatment related response. And it's not terribly surprising that we see this formalin effect. I think there's been some new studies that have come out that show that fixation or preserving samples in RNA later can also have a gene expression type of response. So it's not just formalin fixation that can do this, but other types of sample preservation that can cause these types of effects. But importantly, we're still able to detect our the chemical treatment related response, our response of interest in these samples, and that are having our matched control, so fixed chemical treated and fixed vehicle control, helped prevent us from or counteract that detection of the formalin fixation effect, essentially. So after coming up with all these methods to improve our toxicological samples, and we wanted to see how would this perform with clinical samples. Could we use something like our organocatalyst treatment to get better DNA sequencing results in clinical tissue tumor samples? And because we had seen that the organocatalysts seem to reduce RNA deletion rates, could it apply to DNA? And the original studies that worked with the organocatalysts suggested that it should perform equally as well with DNA as it did with RNA. So we paired up with the National Cancer Institute their biorepository research program where they had human tumor resections that were either frozen or fixed, paired sample design. And then we had FFPE samples and ones where we could apply the organocatalyst treatment for potential to improve them. And in this case, we were also interested in seeing, since the RNA-seq is sort of like the expressed version of the exome, and we were performing exome sequencing for the DNA, could we use the RNA sequencing results as like an added filter or added confidence in some of our variant calls for the DNA sequencing results? And so what we saw initially, just looking at quality scores from variant calls, if you focus on the graph at the top right, and this graph, it looks a little counterintuitive, I'll say, is that, so we have our genotype quality score on the x-axis, and then the cumulative percentage of SNPs detected in those samples. On the y-axis, the solid black line is the frozen, so the results from the frozen sample. And with the shift up that happens with fixation for 12 hours or 72 hours, we had 2 time points, is that really what you're seeing is a higher proportion of your SNPs are at a lower genotype quality score. So higher, shifted higher is bad. And the organocatalyst treatment shifts it down below. So we're getting better quality scores on our SNP detection in our FFPE tissue samples. Surprisingly, they turned out better than frozen, which was an interesting result. But when we look down at the lower graph, now that's focusing on the RNA results, we are not necessarily seeing that improvement. Unfortunately, we weren't able to get just FFP RNA-seq data. We only had frozen and then FFP plus organocatalyst treated, so we couldn't look at that comparison, but it just didn't seem to have any great benefit in this case for at the quality level. And actually, when we looked downstream for SNP detection and other variant analysis metrics, it actually just didn't seem to work out very well. So most of the subsequent slides are just going to focus on the DNA sequencing results and how the organocatalysts impacted the DNA sequencing data. And I will point out an added caveat is with these samples, we only had frozen tumor. We didn't have tumor normal pairs. So some of the contrasts that would have been very interesting to make, we just didn't have access to those samples. So when we focus on just our concordance in SNP detection, so the polymorphisms and variants or known variants in these tissue samples, is that we can look at how concordant were the fixed samples versus frozen. Do we see any genotype changes? Which ones had unique? And then if there were zygosity changes, so that's if we had, when looking at chromosomal pairs, conversions, like heterozygous to homozygous or homozygous to heterozygous. And mainly what we see is this was actually quite interesting, is that we didn't see a lot of genotype changes at all in these samples. And I guess that's perhaps because DNA is more stable than RNA. But when we look at the value of the organocatalyst, is that if you look from the 12-hour fix and 72-hour fix, there's less concordance relative to the frozen in those samples. So, and then when we add in the organocatalyst, we see an improvement in concordance. It's like at least a 10% improvement in concordance of our SNP calls. So it's improving the consistency with frozen in the FFP tissue samples. And as I alluded to, there just weren't a lot of genotype changes in the samples. We did see a little bit more zygosity changes with the in these samples versus genotype changes, but the main genotype change we saw in the fixed tissue samples were these insertion deletions, but it was so small, it was less than 1% of the sample where we saw this. So it was really hard to see like any super beneficial effects of using organocatalysts. So really, I mean, this is an area that could do with some further research into how the organocatalyst is improving these samples. And if we had tumor normal pairs, that would have been really useful for looking at cancer signals in the samples. Those samples weren't available, but we did see some benefits from using the organocatalyst with SNP detection in FFPE tissue samples that could be applied to other clinical type samples. So that was very positive. So after that, this has been now several years since the first 2016 papers came out and testing RNA-seq. During that time, an even newer technology had kind of come to the forefront where it was focusing on targeted RNA sequencing. It's known as TempoSeq. This technology focuses on specific regions of the exome. So it's based off what we know about existing genes in a model organism. So you can't necessarily do discovery applications, but you can look at the known transcriptome and because those probes are specifically designed to regions of the genes and they're short probes, like 50 base pairs, I think, that we speculated that it might be helpful for these really old FFP tissue samples. Instead of trying to reverse transcribe all of this damaged RNA extracted from an FFP tissue sample, what about using targeted RNA-seq to focus in on the specific genes of interest, and how would that perform? And so we were able to go back to these original 20-year-old samples that I talked about earlier and apply targeted RNA-seq, TempoSeq for the frozen and FFPE. And then once we got that data, we reanalyzed the old RNA sequencing data and TempoSeq data together with updated information and aligners and all of that. And we were able to then look at some comparisons between the two in this nice paired design sample. So these are, once again, concordance figures looking at frozen versus FFP. The left is the RNA-seq results split down by each dose level for the DCA experiment. And then the right is the TempoSeq. And you can see what was seen previously is that we just didn't see a lot of genes that were consistent between frozen and FFP, very few, and there's 0 in the low dose group, only 2 in the mid, and then like 5 or 6 in the high. Whereas TempoSeq, we're already having a lot more consistency in frozen versus the paired FFPE tissue samples, higher levels of gene detection. And when you look at TempoSeq frozen versus RNA-seq, since this was a newer technology, we wanted to see how it performed versus kind of the gold standard RNA-seq method. And to the left, we're looking at RNA-seq frozen versus TempoSeq frozen. It's performing very well. There's a lot of overlap in genes across the dose levels. They're quite consistent. The concordance values are very good. And then when we extend our analysis to compare TempoSeq FFPE versus RNA-seq frozen, we still see a nice level of overlap with that response as well. So the TempoSeq seems to be performing quite well relative to the established technology, and FFPE, it's performing better with these really old FFPE tissue samples. In this particular case study where we only focused on liver, but it was very promising. And when we then look at some of those marker genes that we looked at before, CYP4A10 and ACOT1, we can see that the blue bars are representative of the RNA-seq FFPE samples. There's a lot of variability in the response. And yeah, there's just not, I mean, a lot going on. You almost looks, if you focus on the medians, it almost looks like a decrease in expression, and that happens for both of these marker genes. Whereas if you look at the TempoSeq FFPE, which is kind of the pale yellow color, it's showing a response that's much more consistent with the two frozen samples. There's less variability in the response as well. So even now at like individual gene level, it's looking better, at least for marker genes. And overall it's looking better. And then when we actually perform benchmark dose modeling using BMD Express, and we look at, so it was for each of these groups, and then we map those genes to gene sets, and we look at the lowest median gene set as an indicator of that benchmark dose value for the gene set level. The TempoSeq RNA, TempoSeq frozen, TempoSeq FFPE, and RNaseq frozen are all within threefold of the traditional apical benchmark dose value, which was for liver adenoma or carcinoma. So those are quite consistent with each other and with the long-term endpoint of concern. RNaseq FFPE, we still actually were able to get some genes that mapped to gene sets, but there were very few, and it was shifted a lot higher than the TempoSeq results and the RNaseq frozen. So it just looked like at several different levels that TempoSeq was performing better with these really old FFPE tissue samples. And so that then led us to want to look at, is there studies from the National Toxicology Program archive where we could apply TempoSeq data or the TempoSeq platform to get data from some of these really old studies. So this is very preliminary. We went and looked in the archive for studies that had short-term interim sacrifices. So they collected samples earlier on in the study, and they had the multiple dose levels, but then they also had the longer-term pathological data that we could look at for reference. And so that led us to two studies, nickel sulfate and cupric sulfate. This was of particular interest because the nickel sulfate exposure also is an inhalation exposure. So a lot of these proof of principle studies comparing the short-term gene response to long-term apical effects are focused on these oral exposure methods like through the feed or gavage. So this was something that would be interesting to look at. There are some inhalation studies, but not as many. And then we could look at the gene response. And we could also just explore these studies for differential gene expression analysis for comparison purposes, because the collaborators at EPA Duluth who are doing exposures with these chemicals in the fathead minnow. So then we are able to potentially compare rat response to fathead minnow response, look at consistencies, and hopefully then translate potential ecological effects across to our more traditional animal mammal type toxicity values. So this is something that's still very exploratory and preliminary, but when we just look quickly at the benchmark dose analysis of the results, actually when we did the sequencing, there were very few samples. A few samples did have to be removed due to poor pre-alignment, but not a lot, only like 2 or 3 samples that were actually removed because of that. And these studies were actually from like the early 90s, maybe late 80s. So they were quite old at the time, but yet we still get good gene expression results, quite robust. And we were able to map those to gene sets and compare them to the apical responses associated with these particular studies. And what at least we're seeing with this initial analysis, it's still like to redo the cupric sulfate with translating the data to mg per kg per day, but that the gene set benchmark dose value from the short term, so these were like, I think 14 days of exposure, was within 8-fold of the chronic benchmark dose value from the 2-year endpoint. So these were shifted a bit higher than the apical effect, but they're still within, it's within 8-fold. So that was quite promising considering how old these samples are, and have to stay tuned to see what else we might learn about them using these really old tissue samples. So I think hopefully what I've shown you over the period of this webinar is how the FFPE working group has really advanced the use of archival samples for genomics. We've used new technologies like RNA sequencing to improve gene expression. We've characterized factors that influence quality, time and formalin, age and block. We identified the organocatalyst to improve gene expression, not only for RNA sequencing experiments, but also potentially the DNA and SNP detection in clinical samples, so translating our methods to clinical type human samples. And we've looked at potential artifacts that can occur with fixation. And we are now moving more into application with using targeted RNA-seq to get new data from the really old FFPE tissue samples. And so hopefully now I've given you some examples where we have used tissue samples collected from tox studies, as well as human clinical samples. We've identified biomarkers of DCA and DHP exposures as proof of principle, also phenobarbital. Looked at toxicity pathways. We've helped with clinical research and applying the organocatalyst to improve results, DNA sequencing results from tumor resections. And of course, we're applying these methods in proof of principle type experiments to see how well short-term gene set benchmark dose values associate or indicate 2-year chronic adverse effects of regulatory concern. So with that, I'd just like to highlight some nice biorepository resources that are available to everybody out there, in addition to the National Toxicology Program, which is a good archive for gathering information, but there are others out there, including a lot of biobanks. If you are interested in working with human samples, it is difficult. I will say we did learn that the approvals to work with clinical samples were a lot more stringent than getting our tissue samples from the National Toxicology Program archive. But that said, it was still really great to be able to work with those samples and show that we can improve and translate these results to human tissue samples. And with that, I'd just like to say thanks to the FFPE group and also to Susan Hester, who has since now retired from EPA. I don't know how many people know that, but she was a major contributor to a lot of this work, and she is now enjoying her retirement.
Great. Leah, can you hear me okay?
Yep.
Okay. Well, great. We're right at time. We do have one question in the chat, so maybe we'll do that one before we end. Ken Chan asked if there's been any work to look at purely computational approaches to correct for FFPE artifacts, and then shared a recent paper from Nature. Have you heard of any of that?
Oh, yeah, I have. Some people have done that. We didn't do that in particular with our samples, but I do know that people are looking into that. So it's definitely an area that's ripe for exploration. We just in particular didn't focus on that with our methods.
Great. So we are at time. There's one other one in the chat. Maybe I'll email it to you. Sorry, Chris.
Okay.
But just to say thank you so much. We will share the recording. And Leah, would we be able to share your email if there are other questions?
Yeah, that's fine.
Great. Well, thanks everybody. Thanks for the great webinar. Have a great rest of your day.
Okay, thank you.